glue. It acts like a glue. Now, what it does is that it joins the fragments of DNA together, and it's not literal sense of joining. Rather, it helps in forming the bond in between the two fragments. Which kind of bond? So phosphodiester bond is formed yet again so that the fragments of DNA could be joined up. Then comes another enzyme that has a lot of use in the RDT, that is DNA polymerase. Now see, DNA polymerase, it does the process of polymerization. Polymerization means, you can say, forming multiple base pairs to one of the complementary unit that has been given. This enzyme synthesizes a new strand of DNA complementary to an existing DNA template in 5' to 3' direction. For example, it's like this. Okay, this is the DNA sequence that has been given. So tell me what will be made on its complementary strand.
CATCG/3 dash 5.
From, like, for G, what will be made?
C
C. For A?
A
For A?
A ma'am.
A. No, no, I am not talking about palindromic sequence. I'm not talking about that. I'm talking about complementary base pairing. So G pairs up with C. So A pairs up with
T
T, thymine. Then T pairs up with,
A
then
A
C
G
pairs up with,
G
G pairs up with,
C
T pairs up with,
A
and A pairs up with,
T
and C pairs up with lastly
G.
G. So this is a complementary sequence. I wasn't talking about palindrome. When it would be a palindrome, then it will be like that. Okay, the answer that you were giving. So DNA polymerase does this. Like this base sequence that is formed, it is actually formed in the, you can say, the process through the DNA polymerase only. Now today we're going to talk about cloning vectors. Now you people already know from human health and disease that vectors are those substances that sort of like behave like a carrier, right? They are the carrier of a disease in case of human health and diseases. And in the case of recombinant DNA technology, the vectors are those carriers that are responsible for delivering the gene. For example, you have taken out a gene that is a gene of interest. Now you want to put that gene into the host. So how will you do it? You will use cloning vectors for that very purpose, to transfer or to deliver the gene of interest or any foreign gene into the host organism's genome. We need vectors, and that is what a cloning vector is. Now, there are some features or some qualities that the cloning vectors must possess in order to be considered as a proper vector. So, those features of the vectors are, the first thing is origin of replication. Obviously, because we know that the vectors must be able to replicate, meaning it must be able to make multiple copies. So now talking about the origin of replication, so this refers to a particular sequence exactly where the replication will start. Origin means the start of anything. So origin of what? Origin of replication. So this is a part wherein the replication starts, not actually a part, particularly a sequence. Now, whenever any piece of DNA is added to this ORI, that is origin of replication, then because of the replication property, that piece of DNA will be made into multiple copies. So this origin of replication, it helps us to transform the cells and to make the host cell synthesize more and more of the particle of DNA that has been sent. Is this clear to you all?
Ma'am, could you explain the starting part once again?
Yes. See, in any vector, no matter whatever you say, vector A, okay, in any vector, there is a sequence. You people understand the meaning of sequence? DNA sequence, right? So, DNA sequence. So, and in that sequence, we call that particular sequence as origin of replication, because on that sequence only replication starts. What is replication? The making of multiple copies of DNA. Right, that is what replication is. All of you have studied it in molecular biology. So the part wherein replication starts is known as ORI, origin of replication. So what we do whenever we want to put any alien piece of DNA into the vector, we put it or we load it at the part of ORI, so that when this vector will multiply, so the gene of interest will also multiply, so that multiple copies of it would be made. So that is why origin of replication is very, very important. Okay. Now the process of cloning or making multiple copies is done in this way. We use this particular method, that is origin of replication for making multiple copies, and that is what we call as cloning. And what is the heading of our study? It is cloning vector. It won't have double N. It is just cloning vector. Okay, so cloning vector, cloning means what? Making clones, making multiple identical copies. So those vectors that are used for making that are known as cloning vectors. Now, moving forward, the next property that any cloning vector must have is selectable marker. Now, you people pay attention to this. Selectable marker. See, for example, if you people are doing recombinant DNA technology, and you saw a gene of interest. So you used your restriction enzymes to cut it, right? And then you cut the vector also with the same enzyme. Now you have many vectors. Okay, so you took many vectors. And once you have that cut piece of gene of interest, you are adding it to the vector. Hmm? Now you have done it for multiple vectors, and some of them you have not done it for. Now, how would you tell that which one is a recombinant DNA and which one is not? How will you say? How will you know? Like, what do you think about it? I'll tell you, but how would you distinguish between the ones that have been made, meaning modified, or the ones that have not been modified? Hmm? I mean, there has to be some way, right, to identify, because only those ones that have their genome modified are the ones that are important. The other ones are not, right? So the presence of the selectable marker helps us to do that. Okay, meaning the selectable marker, you people understand any marker. See, for example, if you have hundreds of papers. Okay, 100 papers are there, and each of those 100 papers have some different information written. So you have made 10 groups of each of the papers, 10 pile up. Now you want to say that, okay, the first 10 are the important ones, the next 10 are the less important ones, and the next 10 is non-important, the next 10. You have categorized it. So when you have a paper, or you have, let's just say you have 10 groups of those, like 10 sheets, then how would you actually identify which one is more important, which one is less, and which one is least? Tell me your sorting system, normal. How will you do it? This is normal question. This is not related to the chapter. I'm normally asking.
Ma'am, can you answer the question again?
Say the question again. I had such a long question. See, I said that
I got confused in the middle.
you people have a lot of papers. Okay, I'll say 5 papers are there. Okay, 5 papers are there. Out of those 5 papers, some of them are extremely important to you. They are very important documents. Then some of them are very less important. It's like least important. Okay, and then some of them have different categories. Let's just say you have 5 categories of paper. How would you understand, or how would you identify which one is the most important, which one is the least important?
You will read
According
them.
to what is written in it.
Yeah.
So you will take every paper and then you'll read it to find out. But
The
if you are
heading.
in a hurry, what will you do?
I'll put a marker on it.
Like you, how we do? You people have that sticky notes, right?
Yes.
Yeah.
You use the sticky notes. You like make it above the paper so that it hangs. So you can
Hmm.
color coordinate it, right? You can write I for the important one, meaning you will do something to mark it, right? To understand, okay, this file has my important documents. This has miscellaneous stuff. The same thing happens when we talk about the cloning vectors. We should know which one of the vectors are the ones that have important genes in them, gene of interest, and which ones do not. So for that, we use selectable markers. So selectable markers are equivalent to sticky notes. Okay, those colorful sticky notes that you have, these are equivalent to those. Now, the selectable markers, they help us to identify transformants. Transformants are those that have modified gene, meaning they have gene of interest in them. All those vectors that you have put the gene of interest in it, those are transformants, and the ones in which you have not, so those are non-transformants. So because of the help of selectable markers, you can identify transformants, and because of that you can eliminate the non-transformants because they are of no use for you at that point of time. Now, see, there are many genes in the case of prokaryotes that code for antibiotic resistance. See, meaning antibiotic, you people understand, whenever we get sick, the doctor prescribes us antibiotics, right? Why? Because to kill the bacteria, or to kill the, you can say, pathogen. So that works accordingly to kill it. Same way, there are many bacteria that have developed this mechanism that even if you apply antibiotics to them, they will not die. Meaning antibiotics will not have any impact on them. And this property is known as antibiotic resistance. Now, some of the bacteria, they have this in their genes. It's not you can say, acquired mechanism. They have it in their genes. So, to antibiotics such as ampicillin, chloramphenicol, tetracycline, kanamycin, these are the names of antibiotics to which some bacteria have resistance. Okay. And these antibiotic resistance, this gene, antibiotic resistant gene, is used like a selectable marker, particularly for E. coli, that is the most studied bacteria. You should know, like wherever we talk about any bacteria, we always take the example of E. coli, isn't it? So why? Because it is the most like studied bacteria. It has been the bacteria that has been researched a lot. So that is why E. coli. So, what I mean by saying this is that, for example, there is a vector and you have inserted a gene of interest in it. Okay, now you have inserted in it at a point wherein it was giving ampicillin resistance. Okay, it was resistant to the antibiotic ampicillin. Now, if you introduce that foreign gene into that bacteria or into that vector, what will happen is that it will no longer be resistant to ampicillin. Okay. Are you people following me, or is it getting confusing? You're not following me? What is the problem? Tell me, because your faces seem like that.
I'm getting confused in this part. Too many names are there. It's just buffering
Yeah,
in my
it's new. That's why. It's new. That's why, right? And trust me, it's very interesting. It's just new, so it seems intimidating. See, students, I am saying this is vector A. Okay. This is vector B. Try to understand. Now, this vector and this vector both have antibiotic resistance. You people understand antibiotic resistance? Resist, you understand, right? To avoid something, to escape from something. That is resistance, isn't it? To not let anything happen. That is resistance. So both of them have resistance for antibiotics. We know that antibiotics, do they work on humans to kill humans? They work on
Pathogen.
pathogen, and particularly which pathogen?
Bacteria.
Bacteria.
Bacteria. Yes, particularly bacteria. Now we are talking about bacteria only. Okay, and vector in general. So you people understood what vector is, right? All of you have understood
Carrier.
what vector is?
Yes, ma'am.
It's a carrier, and it has been actually taken out from bacteria only. Okay. Now, so I'm saying that bacteria A has antibiotic resistance to ampicillin, which is an antibiotic. Just like how you have Dolo and all of those medicines, they have a name. So ampicillin is also a name of an antibiotic. Ampicillin, tetracycline, these are those antibacterials. They kill bacteria. But bacteria A and bacteria B, they are resistant to ampicillin, tetracycline, all of these things, all of the antibiotics that I've mentioned here. So now I am saying I took a gene. This is a gene of interest, and I put this gene into this vector. Now imagine this was the site that was coding for the resistance for ampicillin. It was coding, it was making certain proteins because of which the bacteria was resistant. Okay, so now I've inserted the gene. Now, because of the insertion of different or foreign gene, this bacteria will not be able to code for ampicillin resistance. Okay, and in bacteria B, I have not put any foreign gene. Still not getting me? Shall I change the language to explain once? Hmm?
Yes, ma'am.
देखो, listen carefully. All of you understand Urdu and Hindi, right?
Yes, ma'am.
Is there anyone who does not? What about the other Siddika, Mariam Lohani? You people understand Hindi and Urdu?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Good. Now see, I am saying दो बैक्टीरिया हैं, ठीक है? One एक है जो कि किसी भी एंटीबायोटिक से बच करके रह सकता है. I mean it will not get affected by it. ठीक है, मतलब वो बैक्टीरिया सॉरी वो एंटीबायोटिक इस वाले बैक्टीरिया पे काम नहीं करे. ठीक है, देयर इज बैक्टीरिया ए एंड बैक्टीरिया बी. बोथ ऑफ देम आर एंटीबायोटिक रेजिस्टेंट. एंटीबायोटिक रेजिस्टेंट मतलब होता है कि दोज एंटीबायोटिक्स विल नॉट वर्क ऑन इट, इट विल स्टे सेफ. ठीक है, आफ्टर दैट अब क्या कर रहे हैं, हमारा जो जीन ऑफ इंटरेस्ट है, वट एवर इज द जीन ऑफ इंटरेस्ट, आपने वो लिया, आपने उस बैक्टीरिया में डाल दिया. अब आपने उसको एक ऐसी जगह पे डाला है विच इज कोडिंग फॉर एम्पिसिलिन रेजिस्टेंस. मतलब ये वाला जो पार्ट है बैक्टीरिया का, ये ऐसा कोडिंग कर रहा है जिससे ये बैक्टीरिया एम्पिसिलिन के अटैक से रेजिस्टेंट है, मतलब बच करके रह सकता है. ठीक है, इस कोडिंग के वजह से. और इस वाले में हमने कोई फॉरेन जीन नहीं डाला है बी में. ठीक है, बी इज नॉर्मल. ए में हमने फॉरेन जीन डाल दिया है. अब उस जगह पे डाला है जिस जगह से हम लोग एम्पिसिलिन रेजिस्टेंस पा रहे थे उस बैक्टीरिया के अंदर. ठीक है, बैक्टीरिया के पास वो रेजिस्टेंस थी, वो पावर थी उस एम्पिसिलिन से बचे रहने के लिए, बिकॉज उस एरिया पे आप वो कोडिंग कर रहा था उस रेजिस्टेंस के लिए. बट आपने यहाँ पे क्या कर दिया, फॉरेन जीन डाल दिया. अब वो कोड नहीं कर पाएगा क्योंकि उसके जगह पे तो फॉरेन जीन आके बैठ गया है ना. तो अब हमने एक प्लेट लिया, ठीक है, इसमें बैक्टीरिया ए और बैक्टीरिया बी को डाला, फिर इसमें बैक्टीरिया ए और बैक्टीरिया बी को डाला, दोनों चीज. ठीक है, मतलब एक बार इसको एक प्लेट में डाला, फिर सेकेंड टाइम दूसरे प्लेट में डाला. अब हम इस प्लेट में एंटीबायोटिक डालेंगे टेस्ट करने के लिए कि कौन सा ट्रांसफॉर्मेंट है और कौन सा ट्रांसफॉर्मेंट नहीं है. तो आई एम पुटिंग एम्पिसिलिन, आई एम पुटिंग टेट्रासाइक्लिन. तो क्या अब मेरा सवाल है कि नाउ विल आई बी एबल टू नो कि कौन सा ट्रांसफॉर्मेंट है और कौन सा नहीं, पता चलेगा मुझे? ये सारा प्रोसीजर से समझ में आ गया टू
Yes,
एवरीवन?
ma'am.
ठीक है, अब मैंने ये ट्रांसफॉर्म वाला मतलब बैक्टीरिया जिसमें मैंने जीन डाल दिया था और दूसरे में नहीं डाला था, दोनों को हमने ऐसे मिक्स कर दिया और दोनों को प्लेट में डाल दिया. ठीक है, बैक्टीरिया के प्लेट में. अब हमने उसमें ये सारे एंटीबायोटिक्स डाल दी, एम्पिसिलिन डाल दी हमने और टेट्रासाइक्लिन डाल दी, दो एंटीबायोटिक डाली मैंने. ठीक है, अब बताओ मुझे, पहली बार मैंने फॉर राउंड 1 मैंने ये वाला डाला, ठीक है, अब बताओ मैं पता लगा पाऊंगी इन दोनों में से कौन सा ट्रांसफॉर्मेंट है या नहीं?
No, man.
Yes.
अच्छा, यस क्यों, नो क्यों, बताओ, वाई यस?
Yes. Yes, because uh, the ampicillin.
I
Uh,
have just put this one for now. अभी forget ampicillin. I have just put tetracycline for now.
what is your question?
My question is, I took both the bacteria after this whole entire procedure. I put it in a plate and I put antibiotic named as tetracycline in it. Now, will I be able to find out which one of them is transformant and which one of them is not a transformant? मुझे पता चल पाएगा हाँ या ना? Yeah, you tell me why was your answer no?
Because A1 had resistance to ampicillin. Ampicillin, right? The name.
You people are not getting me. Listen here. Is there anyone else who understood what I'm saying? Or is there no one else in the class that understood? Anyone that
The first
understood?
part we understood.
This part, your first part, you've understood?
Yeah, yeah.
Then you should have been able to give the right answer, no? If you would have understood. See, I am saying no in the normal case. Okay, in the normal case, A and B, both the bacteria are resistant to all of the antibiotics. Ampicillin, chloramphenicol, you name it, they are resistant to it. Now, whenever I've put a new gene in one of the bacteria, so this bacteria loses the resistance to ampicillin. It is now no more resistant to ampicillin, but the bacteria B is. Hmm? Now I have taken both of these bacteria, put them into a plate and added tetracycline antibiotic. I have added this antibiotic. Now my question is, will I be able to distinguish them?
Yes.
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 How? Tell me how. You have to tell me how.
Because A is not
Because
resistant to tetras
A will be affected and B won't
A
be affected.
be affected.
But why? I am not putting ampicillin here. No, I am putting tetracycline. A lost its resistance to ampicillin, but not tetracycline.
Oh,
Meaning, if I put
Okay.
tetracycline, both of them will survive. None of them will die. But if I put ampicillin now, since A has lost the resistance to it, so A will die, B will be survive. B will be survive. B will survive. Did you people get this?
Yes.
yeah.
Yes, ma'am.
All of you?
Yes.
Okay, good. So all in all, what I mean to say by this is that due to the insertion of any foreign gene into the vector can lose its antibiotic resistance. अगर इंसर्शन वहां पे हुआ है जहां से कोडिंग हो रही है उस रेजिस्टेंस जीन के लिए, इफ द कोडिंग इज नॉट बीइंग डन तो कुछ नहीं होगा, बट अगर हो रही है, इसलिए हम करते ही वैसे हैं कि हमें पता चल पाए कि कौन सा हो गया है कौन सा नहीं हुआ है. ऑल ऑल क्लियर टू
yes.
एवरीवन?
Yes.
Okay. So the first type of selectable marker was antibiotic resistance. All of the prokaryotes have it. Okay, not all of them, but some of them have it. Okay. Now the next type of selectable marker can be color production. See, some enzymes such as beta galactosidase. You people remember lacoperon? Hmm? What was the function of beta galactosidase? You remember that? This lacoperon, you remember? So what was the function of beta galactosidase? No one remembers that. It used to hydrolyze lactose, break down lactose. Anyway, it's no part of discussion over here. I have a different thing to make you understand. See, there are some enzymes such as beta galactosidase that are coded by LAC-Z gene. This enzyme can also be used for selection, meaning it has some property that it expresses which will help us to understand it better. Now see, this enzyme utilizes its substrate, that is lactose, to produce a blue color pigment. Whenever this enzyme is present, a blue color pigment will be made. So whenever we will insert a foreign gene into that bacteria, now it will not make any color. So on the basis of color, you can find out which one is transformed and which one is not. The one that does not have a foreign gene, which will be colored blue or colorless.
It does not have a foreign gene.
Yeah, it doesn't have any gene. I did not put any gene into that bacteria. Will it be blue colored or colorless?
Colorless.
Colorless.
Colored, right? Why? Because in that, the beta galactosidase will do its work normally. But I took a bacteria and I put the gene in it now. Now that will not make any color. So out of the two selectable markers that we had discussed, antibiotic resistance and this color production, which one do you think is better in terms of lab work and why?
Color,
Color one. The second one.
Okay, why?
because it is easier to
Because
identify.
It is easier to identify, right? You can see it and then you'll know. But for that one, you have to do so many plating. First, you have to put ampicillin, tetracycline, whatever is the resistance, right? So this one is better. So you people understood selectable marker? All of you understood? Every single student in the class?
Yes.
Yes,
Okay,
ma'am.
good. See, there are a lot of technicalities in this chapter. So as long as you are paying attention, no issues. Now talking about the next property or the next feature that the vector should have is the presence of cloning site. Obviously, for example, I'll take a very silly example. For example, for girls, there are so many hair accessories available, right? Clips are there, hair bands are there, hair extensions are there, isn't it? So any girl that is buying the hair clips or clutches or anything of that sort, she can only use it if she has hair. If the girl is bald, is it of use to buy any clips? It's not, right? The same thing. If there won't be any site where you'll put the gene, is the vector useful to you? It's not. Where will you put the gene? Because the whole thing is revolving around that only. So cloning site is the site where the alien gene or the foreign gene is linked at. This is the site wherein you take the gene of interest and you link it at that. Now the number of cloning sites, see, we can say that one vector can have multiple cloning sites, but that's not ideal. The presence of too many cloning sites is not ideal. Our vector should have one cloning site, because if there are many cloning sites, then again it will be very complicated for us to understand where did we put it. Did we put it at site 1, site 2, site 3? We'll never know. So ideal condition is that you have one cloning site in the vector, because more than one cloning site will complicate the process of cloning. Cloning sites are clear? Then comes the size of the vector. So this whole entire thing that I'm talking about, can you do this in humans? Can you do gene modification in humans? Give me two reasons. That's right. You people, all of you are right. Those who nodded their head like this, you are right. Now tell me, why can't you do a gene modification in humans?
Because it's not allowed.
Very good. The most basic and the most important reason, it's not ethical. You can't, like, what will you do? You go on the road and you bring the person and start taking out his DNA. You can't do that. So first reason is that. And secondly, you can't do it because humans have a larger lifespan. We replicate differently, isn't it? So that is why we can't do. Now, all of this recombinant DNA technology that we are discussing, where do we do it? In which organism?
Lab rats?
No,
No.
no, 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 not lab rats. Lab rats are
Micro
also genetically modified, but these vectors, the creation of vector, all of this, where is it done?
microorganism?
Prokaryotes, right? You bacteria, microorganism, correct, but bacteria. So the size of the vector should be so tiny because we have to incorporate in the bacteria. So the size of the vector should be very small as large molecules tend to break down during purification. So small size is favorable. There is one more thing I have forgot to mention it here. They should have high copy number. What do you understand by this, students? High copy number.
They should be They should be able to replicate into a bigger number, like a larger amount.
Yes. They should be able to replicate a lot, right? So that is, like, these are all the features that a particular cloning vector must have. Any doubts? Now let us see some of the examples of a vector. Examples of vector. What are those vectors that we use? Now, up until now, we had discussed about general vectors, right? Now we'll talk about the examples of vectors that are used commonly in the RDT. The first one, the most important one, is plasmid. Have you people ever heard about plasmid before? Where? I think all of you have, isn't it? I don't think there will be any student who did not hear about plasmid before. Is there anyone? If there is anyone, you can say it's all right. It's no biggie. No one is saying. Means everyone has heard. So now it brings me to the question, where have you heard it? Where have you heard about a plasmid? And I'll give you one hint, it's not in class 12. While you people were studying the chapter cell, in that you might have heard about plasmid in prokaryotes. Meaning in the prokaryotic cell, there is a particular region wherein the DNA, or you can say if this is a bacterial cell, in the bacterial cell, we know that there is a region wherein dense chromatin material or whatever structure the genetic material of bacteria is. This part is called as nucleoid, right? Nucleoid. Now they have a circular disc-like structure present in them, and that is what plasmid is. So most of the bacteria, they do have plasmids present. Now let us see what are plasmids. Look at the various adjectives used to describe them. They are extra chromosomal meaning not connected with the chromosome, normal chromosome. So they are extra chromosomal, they are circular, as I showed you in the diagram right now. They are circular, they are not essential. The bacteria does not need it to survive. Apart from that, they are double stranded. Just like how I've shown you, two circles, concentric circles, that means that they are double stranded. They are autonomous. What is the meaning of autonomous? Can anyone tell me? Autonomous. Autonomous means it does not have any regulatory body. It can regulate on its own, meaning it regulates itself on its own. Then comes self-replicating pieces of DNA. So many adjectives to describe plasmid. Extrachromosomal, circular, non-essential, double-stranded, autonomous, self-replicating pieces of DNA that are found in bacteria and some yeast cells. Yeast is a what? Yeast is what? Which kingdom does yeast belong to? Student, these are very basic things. Yeast is a dash. What is it? Yeah, what are you saying, Mariam? Say. You were saying something. Yeah, yeast is a fungus. Very good. Exactly. Yeast is a unicellular fungus. All right. Now, these plasmids are those structures in the bacteria that helps them to have that antibiotic resistance property. The property that we have been discussing for a while. So that property is provided by plasmid in the case of bacteria. Now see, this is the most famous picture of a plasmid. It is the plasmid of E. coli. Okay. And remember that a plasmid is a cloning vector. If I say plasmid, if I say cloning vector, it's the same thing, right? You people understand this much or not? So see, don't get so confused by all of the labelings that the diagram has. You people have to learn it, but don't get confused or scared. Now, if you look at these, SAL1, BAM, BAM H1, HIND3, I wrote about that below because I was going to ask you a question, so I hid it. These all, have you seen it before? To the
Yes,
students that
ma'am.
were present in the previous class, they'll know. Yes. Where have you, you left the class. Where else have you seen the other students? Tell me, where have you seen these words? Have you seen it before? Yes or no? Amna, have you seen it before? Amna, say, have you seen it before? Say yes or no. Say something. Am I audible, Amna? Am I audible to you all students?
Yes, ma'am.
Okay. I don't know. Her screen is frozen or something. Yes, tell me, Yaya, where have you seen these?
They are restriction enzymes.
Yes, correct. They are restriction enzymes. Right? You might have seen in the whole long list that I provided for the restriction enzymes. Now see, all of these restriction enzymes, they have certain properties, or these are the, you can say they are provided or they are present in a plasma. Now, HIND3, ECORI1, BAMH1, SAL1, PVU, all of these are like restriction sites also. These are the site of cloning as well. Okay. Meaning, if we are using one of the restriction enzymes such as HIND3, okay, if you are using hind 3 is available outside also, because you are using it to cut the foreign DNA also, right? So you, what I said as a thumb rule, it is important to remember that the restriction enzyme which will be used for cutting the vector should also be used for cutting the gene of interest. It should be same, or else you can't attach it. So if I say hind 3 has been using, we are using hind 3 for cutting. So this hind 3 will cut here. It will not cut here or here or here. It will cut at this point. Getting my point? That is why different points have different restriction enzyme. That's it. Nothing too complicated. But you have to remember the labeling. So on right hand side, which one is present? On left hand side, which one is present? Now, the ORI, you people already know. This is the part. This is where origin of replication, meaning this is the part where replication starts. Okay, in the bacteria. The replication does not start anywhere else, only here. Then AMPR, AMP with an R on it, TET with an R on it. It means ampicillin resistance. It means tetracycline resistance. The R stands for resistance. So for example, if I load a gene of interest at BAM H1, so what will happen? If I load the gene of interest at BAM H1 site, what will happen? What will happen? I just taught you. Nothing will happen. Yes, Mariam, say.
It
It.
it won't be resistant back
Yeah,
antibiotic
which
resistant.
one? It won't be resistant to which antibiotic?
Tetracycline.
Tetracycline. Correct. Exactly. That is what will happen. If I load the vector at PVU1, then it will lose the ampicillin resistance. So that is what will happen. This is the whole thing I taught you people in 2 languages, and still you people don't understand. Now you do, right? All of you do. Then there is another thing, that is ROP. If you see here, ROP. See, this is region of plasmid. What happens here? This region codes for the proteins that are involved in the replication. So ROP will help ORI to replicate. That's it. So this plasmid has a name. It is PBR320. It has a name. Okay. Any doubts? So I told you that plasmids, they are extra chromosomal pieces of DNA that are found in bacteria, isn't it? Now, normal chromosomal DNA is also found in bacteria. So we are going to see what is the difference in between them. Plasmid DNA and chromosomal DNA. You should know. This is just like a knowledge part. See, both of them are double strand. That's not a difference. But plasmid DNA is always circular like this, double stranded and circular. Okay. But chromosomal DNA can be circular or it can be linear like that. Okay. The plasmid DNA is present without histone proteins. Histone proteins are absent in it, and the chromosomal DNA has histone proteins present. Histone proteins are important for wrapping of DNA. And plasmid DNA, like I told you, it's non-essential, meaning it does not carry any important gene. It only replicates, it only divides. Chromosomal DNA, obviously, all of the properties that the bacteria have, it is because of the chromosomal DNA, right? The DNA that is present on the chromosome. Lastly, it replicates independent of main genome, meaning it doesn't require any regulatory body. Like I told you, it is autonomous. But chromosomal DNA is not autonomous. It replicates with the genome. Clear? So the differences between plasmid and chromosomal DNA is clear to you all? Any doubts up until now? Selectable markers are clear? Okay. Now you people can note down, and see, while you're noting down, you have to draw this. Okay. All right. We have to start from DNA ligase, isn't it? You people wrote down till here the remember part. Did you write this part?
Yes, ma'am. We even wrote DNA ligase. We have to start from that.
You have to start from DNA polymerase, right?
Yes, ma'am.
Okay.
Done, ma'am.
Done, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am. Done, ma'am.
Done, ma'am.
Done, ma'am.
Everyone's done? All of you are
Yes,
done?
ma'am.
Okay.
Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am.
Done, ma'am. Done, ma'am.
All of you are done?
Yes, ma'am. Yes, ma'am.
Done, students?
Yes, ma'am.
No, ma'am. 1 minute, ma'am.
Okay, okay.
Done, ma'am.
All right, so we'll keep it till here only for today's class, and in the next class, we'll continue.